For most of the 20th century, geneticists told a simple story about eye color. Brown was dominant. Blue was recessive. You could predict a child's eyes with a chart on the back of a textbook. And every shade that wasn't brown, they said, traced back to a single mutation somewhere in Northern Europe. Case closed, except it wasn't. Because when researchers finally decoded the DNA of ancient populations buried beneath the oldest crossroads on Earth, they found something the textbooks never predicted. The Middle East, the region most associated with dark brown eyes, was carrying one of the most complex eye color signatures on the planet. And the color at the center of that mystery was hazel. To understand why this matters, you have to see what scientists believed they already knew. Every human being alive 10,000 years ago had brown eyes. That much is agreed upon. Brown is the ancestral state, the product of high melanin concentration packed into the front layer of the iris. Roughly 70 to 80% of the world's population still carries that default today. Blue eyes, the story went, came from a single founder mutation, a tiny change inside a gene called HERC2, sitting on chromosome 15. That change flipped a regulatory switch. Not a switch that makes pigment, but a switch that controls a neighboring gene called OCA2, which manages how much melanin your iris produces. Flip it one way, melanin flows freely. Brown eyes. Flip it the other way, and OCA2 goes quiet. Melanin drops. The iris lightens. Blue. In 2008, a team led by Professor Hans Eiberg at the University of Copenhagen published the definitive study. They examined 155 blue-eyed subjects from Denmark, 5 from Turkey, 2 from Jordan. Every single one carried the same haplotype. One mutation, one common ancestor. One origin story, most likely a single individual who lived near the Black Sea between 6 and 10,000 years ago. The paper landed like a verdict. Blue eyes were a European trait. Brown eyes belonged to everyone else. And intermediate colors, green, hazel, amber, were treated as minor variations. Statistical noise, blending artifacts produced when dominant and recessive alleles didn't sort cleanly. Hazel was treated as a minor variation. Hazel, in particular, was overlooked. Geneticists studying pigmentation in European cohorts rarely gave it its own category. It was lumped with brown in some studies, folded into a vague intermediate bin in others. No one paused to ask a simple geographic question. No one plotted where hazel was most concentrated on a map. That was the mistake. Because if you mapped hazel eye frequency across the globe, a pattern would emerge that the textbooks never mentioned. 5% of the world's population carries hazel eyes. But in Lebanon, that figure climbs to somewhere between 15 and 25%. In Turkey, 10 to 18%. In Iran, 8 to 15%. In Syria, 12 to 20%. The global average is 5%. The Middle Eastern average is 2 to 5 times that. These numbers do not distribute randomly. They concentrate in a geographic arc stretching from the eastern Mediterranean coast through northern Mesopotamia into the foothills of the Zagros Mountains. The same arc, it turns out, where 3 of the most genetically distinct ancient populations in human history collided. So why did science miss this for so long? 3 reasons, and each one compounded the others. First, the model was wrong. For decades, eye color genetics was taught as a single gene, 2-allele system. Brown dominant, blue recessive. A high school Punnett square could handle it. But that model cannot produce hazel. It cannot explain how 2 brown-eyed parents have a hazel-eyed child, or why the same iris can look green under fluorescent light and gold in afternoon sun. We now know that at least 16 genes contribute to iris pigmentation. They interact through epistasis, incomplete dominance, and long-range regulatory feedback loops that span thousands of base pairs on chromosome 15 alone. The old model didn't just oversimplify, it made hazel genetically invisible. Second, the sampling was biased. The landmark studies on eye color genetics, including the Copenhagen founder mutation paper, drew their subjects overwhelmingly from Northern European populations, Danish cohorts, Dutch biobanks, Scandinavian registries. When Middle Eastern populations appeared in the data at all, they were used as controls, brown-eyed baselines against which European variation was measured. Nobody sequenced the Lebanese grandmother with the golden ring around her pupils. Nobody asked what was happening inside those supposedly homogeneous brown-eyed populations. Third, and this is the reason that locked the error in place for the longest, there was no ancient DNA from the right time and place. Without genomes from the deep past of the Levant and Mesopotamia, researchers had no way to trace the admixture event that created the Hazel signature. They could see the trait in living populations. They just couldn't see where it came from, or when, or how. That changed in 2018, and it changed because of the cave. Peki'in Cave sits in the limestone hills of Upper Galilee in northern Israel. It was first excavated in 1995 by archaeologist Dina Shalom, when workers discovered a massive underground necropolis hidden behind a natural rock seal. Inside, roughly 600 individuals had been buried during the late Chalcolithic period, around 4,500 BCE. The burials were unlike anything previously seen in the region. Ornate ceramic ossuaries, some standing over a meter tall, were shaped into stylized human faces with wide, staring eyes and prominent noses. The pottery was spectacular. The metalwork included objects created with copper technology traced to eastern Turkey or the Caucasus. None of it matched earlier or later material cultures in the Levant. It appeared, flourished for about 600 years, and vanished. For 3 decades, the central question haunted every researcher who studied it. Where did these people come from? In 2018, a team led by Eyadawan Harney and Professor David Reich at Harvard University, working alongside Tel Aviv University researchers Dr. Hilla May and Professor Israel Hershkovitz, finally answered that question. They extracted bone powder from 48 skeletal remains. From 22, they reconstructed viable genomes. The preservation was extraordinary, likely thanks to the cave's cool interior and a limestone crust that sealed the bones like a biological time capsule. What those genomes revealed changed the entire picture. The Peki'in people were not entirely local. Their ancestry modeled as roughly 57% Levantine Neolithic, the descendants of Natufian hunter-gatherers who had lived in the region for millennia. But 26% of their genome matched Anatolian Neolithic populations from what is now Turkey, and 17% came from Iranian Chalcolithic groups in the Zagros highlands. 3 genetically distinct populations that earlier studies showed were as differentiated from one another as modern Europeans are from modern East Asians, had fused into a single community, in a single cave, in a single generation's worth of burials. Before we see what the pigmentation data from these genomes reveals, if you're finding this genetic detective story as fascinating as we do, subscribe so you don't miss future stories like this one. Here is where the signature appears. Among the Peki'in remains, 49% carried the allele associated with blue eye color. This variant had been completely absent in earlier Levantine populations. It arrived with the northern migrants. But blue eyes were only half the story, because Hazel does not work the same way as blue. The genetic architecture that produces Hazel requires a moderate reduction in OCA2 expression, not a full shutdown. And one key variant that achieves exactly this is a missense mutation called RS180-407, a single nucleotide change in exon 13 of OCA2, which swaps the amino acid arginine for glutamine at position 419. This variant doesn't silence melanin production the way the blue eye switch does. It dims it, just enough to shift the iris from solid brown into that layered, shifting territory of green, gold, and amber that we call hazel. The melanin doesn't disappear. It redistributes. It concentrates near the pupil, and thins toward the outer rim, creating the characteristic sunburst pattern. And then physics takes over. Light enters the iris, hits the low melanin zones, and shorter wavelengths scatter backward. Blue mixes with residual gold pigment. The eye appears green. Move the light, shift the angle, and the ratio changes. Green becomes amber. Amber becomes brown. No pigment has moved. Only the light has changed. Now the three proof beats lock into place. First, what the evidence shows. The RS18T407 Hazel green allele is found almost exclusively in Europe, Southwest Asia, and Central Asia, reaching up to 11% frequency in certain populations. It does not appear in East Asia. It does not appear in sub-Saharan Africa. Its geographic fingerprint maps precisely onto regions where ancient Anatolian, Caucasian, and Iranian lineages mixed with local Levantine and Mediterranean groups during and after the Chalcolithic period. Second, what it rules out. This is not random variation. Hazel eyes do not appear uniformly wherever brown-eyed populations exist. They concentrate where specific ancestral streams overlapped. The RS18T407 allele operates through a different mechanism than the blue eye switch. It is not a diluted version of blue. It is a separate genetic event, carried into the Levant by a separate migration, producing a separate phenotype. Third, what it implies. Hazel eyes in the Middle East are not a modern anomaly. They are not the artifact of Crusader-era contact, Ottoman admixture, or recent European gene flow. They are a signature roughly 6,000 years old, written into the genome during the Chalcolithic period, when the greatest population merger in Near Eastern history fused three ancient lineages into one. The Natufians brought the ancestral brown eye baseline, a genome adapted to thousands of years in the Levant. The Anatolian farmers brought light pigmentation variants that had been evolving independently since the last ice age. The Zagros Highlanders contributed their own distinct genetic layer. And in the narrow arc where these three rivers of ancestry converged, the precise combination of regulatory dimming and structural light scattering that we call Hazel emerged at frequencies found almost nowhere else on Earth at that scale. But not every researcher reads the data the same way. Some caution that Hazel prevalence estimates in the Middle East rely heavily on self-reported surveys, not standardized iris photography, and the boundary between light brown and true hazel is notoriously subjective. Others note that RS18T407 alone explains only a fraction of intermediate eye color. Additional modifiers, perhaps population-specific polymorphisms, may play roles not yet mapped. A study of populations along the ancient Silk Road found that individuals carrying identical HERC2 haplotypes had significantly different probabilities of developing green or gray irises, depending on whether they lived in the Caucasus or Central Asia. The same genetic hand, dealt across different backgrounds, produced different results. Something else is at work. And then there is the question of selection. The blue eye haplotype shows strong signatures of positive natural selection in Europe, meaning something, whether sexual preference, improved light sensitivity at northern latitudes, or melatonin regulation in dim winters, actively favored its spread. The Hazel variant shows no such signal, no selection pressure, no survival advantage. It appears to have drifted, carried passively by population mixing, never pushed by any evolutionary force. A study at Yale University, which genotyped over 3,400 individuals from 72 populations for 21 SNPs across the OCA2 HERC2 region, found that while blue eye haplotypes showed strong extended haplotype homozygosity, the Hazel-associated RS18T407 variant did not reach significance. The trait was preserved not by biology's hand, but by demography's inertia. Which raises an unexpectedly profound question. If Hazel was never selected for, then its persistence across 6,000 years of invasion, empire, and population turnover is itself the evidence. It survived not because it was useful, It survived because the three populations that created it never fully replaced one another. Layer after layer of conquest washed over the region. Assyrians, Persians, Greeks, Romans, Arabs, Crusaders, Ottomans. And through all of it, the underlying genetic architecture held. Today, if you walk through Beirut, or Istanbul, or Tehran, and catch the light shifting across someone's iris, green to gold to brown in the time it takes them to turn their head, you are watching an optical event 6,500 years in the making. There is no green pigment in that eye. There is no gold pigment either. There is only melanin, arranged in a gradient that a Natufian ancestor could never have imagined, shaped by Anatolian farmers who carried seeds and light eye alleles southward across mountain passes, refined by Zagros Highlanders, whose contribution we can now read in the letters of a single gene. Light enters that iris, and physics does the rest. Short wavelengths scatter backward through low melanin tissue. Blue mixes with residual amber. The eye appears green. Shift the angle, and green becomes gold. The color has not changed. Only the observer has moved. If this hidden chapter of human genetics intrigued you, subscribe to explore more mysteries the evidence is only now revealing. Roughly 400 million people alive today carry hazel eyes. But in the arc from Beirut to the Zagros, the concentration remains 5 times the global average. A living fossil of the moment 3 separate branches of humanity converged into one. If hazel eyes gave no one a survival edge, were never selected for, never served a biological purpose, how did they hold on through 6,000 years of empires, migrations, and wars while so much else was erased?